Now the second thing is the composition of the CSF and how that composition changes in various pathologies and the biomarkers. So we are going to go through those now. So here we have done the production and the barrier. We'll do the composition now. We've done the flow. We've done the reabsorption. So let's do composition of CSF, profiles of CSF, and the CSF markers. Okay guys, so the CSF profiles and the composition. Let's see this. Again, you would see in this video, you'll see a little card as well that shows you various profiles. Let's go through those here as well, that how does the CSF change in various pathologies. So let's start with the simple ones. Let's say bacterial meningitis, bacterial infections. So what happens is bacteria love to eat glucose. So what will happen to the glucose level? Glucose level will go down. Simple. Proteins. Bacteria can cause inflammatory reaction. So the inflammatory reaction would have proteins, most specifically C-reactive protein will be found in the bacterial meningitis, plus the cells, the neutrophils will come in and try to combat the bacteria. So that local damage that would occur to the neutrophils and to the bacteria and the other cells around, that would cause the protein increase. So protein will increase. How about the cells? So who handles the bacteria? Mostly neutrophils. So neutrophil levels are increased, protein levels are very high, glucose level are down. Suspect bacterial meningitis or infection. Now one more thing here, tuberculosis is actually bacterial, right? However, remember tuberculosis is handled slightly differently compared to the rest of the bacterial infections. So instead of the cells being neutrophils, the cell are going to be lymphocytes. Now lymphocytes are the cells that would also be increased in viral meningitis. So this is what you have to be as a doctor or a student or as a nurse. You have to make sure that you keep that in your consideration, that the lymphocytes are increased instead of neutrophils. This is a bacterial meningitis. So not all bacterial meningitises will have neutrophils. Here is the case, TB would have lymphocytes. How about proteins? Again, proteins, yes, increased or not. Glucose will be reduced because we have a bacterial meningitis. So that is the TB, which is also bacterial meningitis, but the profile for the cell is different. The profile is like viral. Then fungi. Similar structure that like the bacteria. However, fungi are not eating glucose, so not much change here. The inflammatory proteins present or not present, so maybe normal to high but not too much. So I'll just make a tiny little arrow here, but most important thing, monocytes will be present as the cells that are handling the fungi. Monocytes. Then viruses. So what do viruses do? Eat glucose? No. Make proteins? No. Will they cause inflammatory reaction? Yes. Can that make proteins? Yes. Will they have antibodies produced? Yes. So there is a chance of increasing moderately the proteins. So in viral meningitis, it's not necessary that proteins are always elevated, they could be moderately elevated. However, the cells will be present, lymphocytes. So I'll just make again over here a tiny arrow for the protein, meaning normal to moderately high proteins would be present, no change to the glucose. Then we go to something that is interesting, autoimmune diseases. What do you think would happen here with the autoimmune diseases? So look, autoimmune diseases, is that disease going to eat glucose? No. Is it going to create proteins? Not really. However, because it is an autoimmune disease, because the antibodies will be produced, those antibodies may be causing protein level to increase. However, there is going to be another test that you need to do. The tests will be, you have to do the oligoclonal antibody profile, you have to see the synthesis rate of the antibodies, and you have to see the IgG indexes. So those indexes are to be seen. So number one, oligoclonal antibodies, IgG index, and then the rate of synthesis. What does that mean? Rate of synthesis would tell you how fast the antibodies are being produced and how much damage they would be causing. What about the cells? Of course, we are talking about the autoimmune diseases, so it is the lymphocytes. Neoplasms or cancers, tumors. Neoplasms, again, would they do anything to the glucose? No. Can they increase protein? Yeah. In neoplasm itself can be the cells that are destroyed and that are being produced. So they could produce the proteins, then inflammatory reaction could be present, that would produce the proteins, and so on. So proteins, again, a tiny little arrow here, and then lymphocytes, of course, the cells of the immune system trying to get rid of the neoplasm. And final thing here is trauma. So in the case of trauma, the only thing you would see is RBCs present. Proteins sometimes can be increased if there is the inflammatory reaction that has started. Glucose, no change here. Now one thing, that if you only see the RBCs present in the very first sample and not afterwards, then that is a lumbar puncture anomaly and has nothing to do with the, there is no other trauma present. Okay, so the last topic for the CSF, so we've just done the composition and the CSF profiles. Now we are going to do the biomarkers. So the biomarkers, the ones that are interesting are number one, SB100, then tau proteins, and then beta amyloids. A beta amyloids. So let's very quickly talk about them. SB100 is present in the astrocytes. It's made by the astrocytes. It's a group of proteins. So what do you think? Whenever the astrocytes will be damaged, SB100 would spill out in the CSF and you'll be able to find it. So normally injury to the brain is marked by the presence of SB100 inside the CSF. So that's one. Tau and A beta amyloids, they are present in the Alzheimer's. We have done the Alzheimer lecture separately. You know that when the gamma secretase appears and cuts the APP protein incorrectly, that produces the A beta amyloid. However, that beta amyloid goes and makes a plaques. So reduction in beta amyloid, increase in beta amyloid in the plaques is a marker of Alzheimer's. And finally the tau, remember that tau proteins are the proteins that take part in making the microtubules in the neurons. When they get extra phosphorylated because of the presence of the beta amyloids, then those phosphorylations cause the tau proteins to be degraded or become, more polar. When they become polar, they start causing the cell membrane disruptions, which then start causing the neurons to be damaged. So tau protein increase or A beta amyloid reduction is a marker for Alzheimer's. Thank you very much.